with Rachel Brouwer of the ENIGMA-Plasticity working group. And in that working group we're interested in longitudinal changes over time. So how much does one person change in their brain during their life? And we want to investigate what genes are involved in these brain changes over time. What has been studied before in our department, but now we have shown worldwide, is that the amount of change that people have in their brain over time is driven by their genome. There are two pools of genes involved, one on volumes themselves and one on the amount of change over time. And these are separate pools of genes. So that means that there are genes that are specific for brain plasticity. Now that we know that genes are involved in brain changes over time, we first want to know what genes. And then secondly, we want to know whether the genetics involved in aging is the same as the genetics involved in development. And those could be different, but those could also be the same process that acts on a continuum throughout your whole life. And when you say development and aging in the same sentence, you forget what lies in between, and that is healthy adulthood. And that is a period that is not very interesting in terms of brain changes at a group level, because at a group level that doesn't seem to change much. But if you look at the individual, there are people that are growing still till late in adulthood and other people that start to decline earlier. So even in adulthood there are individual differences in brain changes, and those could be very informative as well. So what we are wondering is whether this is all part of the same process, that over life there is some genetic factor that makes sure your brain stays healthy, in healthy development and healthy aging, or that you're unlucky and have a gene pool that makes your development less efficient and then aging maybe faster. So if we are going to look for the genes that are involved in these brain changes, we also want to know whether these are different genetic processes involved in development and in aging. Well, the big change that we are doing compared to some of the other ENIGMA groups is that we look at longitudinal measurements, so the same people measured twice. We have looked at twin samples that were all measured twice to investigate the genetics of the changes. And what you basically do is look at monozygotic twins. Those are genetically almost identical, and dizygotic twins that share 50% of their genes. And if the monozygotic twins are much more alike in their change over time than the dizygotic twins, that is an indication that genes are involved, explaining variation between people regarding the brain changes. For now we have been working with healthy people only, but we plan to expand that to diseases, especially since psychiatric diseases are sometimes characterized by deviant developmental patterns rather than differences between control subjects and patients at one time point. It's not that people are that different on one measurement, but if you follow them over time, you see they behave different than healthy controls. So in that sense, they could give more insight in the genes that are involved in brain changes over time.